Assalamu alaikum, everyone. My name is Abdulrazak Qatti. I'm a third year medical student. And inshallah, we are having a glycogen metabolism lecture. It will be fun and easy lecture, inshallah. Let's start. So, can you guys tell me what's glycogen, or what do you know about glycogen? Like, glycogen is the storage source of the energy in our body. In simple form, it's the glucose monomers or the sugar glucose that is associated with each other, and they actually form the glycogen. In this lecture, we will know how these glucose monomers form and what are the linkage between each of them. And we will discuss how can we break these linkages in order to give glucose to our body. Tamam. These are the learning objectives. We will start by glycogen synthesis and breakdown. Then we will know how hormones can regulate the glycogen metabolism. And then we will discuss some diseases like McArdle and Bomby diseases. Like, we know that glucose is the primary source energy for our body. We have some structures like the brain and the RBCs that utilize on glucose. Why is the RBC utilizes only glucose? Because it does not have mitochondria. Thus, it cannot use oxygen. Not only our brain and RBCs, but we will have also exercising muscles. What does that mean? It means our exercising muscles use two things. تمام? When it's light exercises, it will use oxygen and it will give us ATP. But when we are having intense exercises, or for example, there is a lion who's chasing us, the muscles will contract intensely, and this will be a heavy exercise on our body. So the oxygen will not be enough for our muscle to give ATP. Instead, it will utilize glucose to give us ATP. This is called anaerobic oxidation. طيب. The primary source of glucose is three things. The first one is diet and degradation of glycogen and gluconeogenesis. So guys, which one of these do you think is the fastest way to get glucose? Is it the diet or degradation of glycogen ولا gluconeogenesis?
Degradation of glycogen.
تمام. Okay, correct. Why? Because if there is a lion which is chasing you, of course you will not start eating, or your body will not start making glucose. It will instead degrade glycogen as a fast way to give the body the energy source. طيب. We said gluconeogenesis is a slow process, so we need something called glycogen. And yes, in absence of oxygen or any other energy source, we will use muscle glycogen. Now, there is important concept, which is the muscle. This is the muscle, as an M here, and this is the liver. تمام. We know there is a lot of muscles in our body, and sometimes we need heavy energy and a lot of glucose. So our muscle will have its own glucose or glycogen. It will not share, معلش, it will not share any of the glycogen it has to the other body. Why? Because it needs a lot of energy and glucose. Thus, if there is a lion chasing you, it will use all of the glycogen. Of course, it will not get the glycogen from the blood, because that will be a rapid depletion. تمام. and the liver will give the glucose to all our body. So there is two main source or storage organs in our body, the muscle and the liver. And please remember this because it's so important. Okay, تمام. The glycogen in our muscle, the muscle is so selfish, it will not give glycogen to the body. And this is because there's something called in our cell, the glucose will be trapped. This is glucose. It will be trapped if it's associated with phosphorylation, تمام? And it will be free if it's not associated with phosphorylation. So this is phosphorylation minus, which is not phosphorylated. This is free. It can go out of the cell, and this cannot go out of the cell because it's phosphorylated. Now the muscle cannot dephosphorylate the phosphorylation. So all of the glycogen and glucose will stay in the muscle, while in the liver, it can go outside. The glucose can go outside and provide the glucose for our body. And why the muscle cannot dephosphorylate the enzyme? Because it's like something called glucose 6-phosphatase. Phosphatase, it means hydrolyzation, which means we are removing something out of the glucose 6-phosphate, which is the phosphate. تمام.
Yes.
We're done with that. We will start with glycogen synthesis, which is how the body will make the glycogen linkage and the glucose linkage between each other. But first, we should understand the branching system of the glycogen. This is glucose. The blue thing is glucose, and the white thing is the linkage. We have two types of linkages. The first one is linear linkage, which is this one, which is a straight line. We call this linkage carbon 1,4, which is this one. And we have another linkage, which is called 1,6, carbon 1,6, which is in a branch form like this. So this is called alpha 1,4, and this is called alpha 1,6. And each of these will have its own enzyme that will make the linkage between each other.
Hi, can you repeat the last 3 slides because something happened to my computer. I'm sorry about that.
I should repeat the last 3 slides?
Yes, please.
Okay, Tama. Sure.
Sorry.
No problem. So what I was saying here, the muscle will not cannot give glucose and to the body because it lacks something called glucose 6-phosphatase. 6-phosphatase. This means this enzyme will remove the phosphate out of the glucose, so the glucose monomer can go freely to the blood. While the liver, it have it, so the glucose can easily go to the blood. This is why the muscle cannot give the energy to other sites on the body. So we can consider it as a selfish organ. So we're done with that. Let's start with the glycogen synthesis, which is how we're gonna synthesis and make the linkage between each glucose. We have two types. The first one is alpha 1,4 and alpha 1,6. The linear form or the linear linkage, which is a straight line, we call this alpha 1,4, and the branching linkage we called alpha 1,6. And each of them will have its own enzyme that can make the linkages. طيب, how can we make the glycogen synthesis, or what is the first step that tells the body that we have to make glycogen? Did you guys take the glycolysis lecture?
Yes.
Okay, so what's the first step in glycolysis? It's
breakdown of glucose to fructose 1,6-bisphosphate.
yes. So after that, we will have glucose 6-phosphate, and when the body sees that there is a glucose 6-phosphate and we need to make glycogen, it will convert glucose 6-phosphate into a glycogen, and it will prevent it from going to glycolysis. So again, the first step in making glycogen synthesis, or the thing that tells the body we have to make glycogen, is glucose 6-phosphate. Yeah, this is the same thing, but the new thing in this slide is we have something called glycogenin. Glycogenin is a protein that's the same as infrastructure of everything. The body will give the glucose 6-phosphate to glycogenin, and then the body will start making glycogen. So there's two important steps. First thing is the availability of glycogenin, which is the core protein, and then it will adhere to glucose 6-phosphate, and this will start making like glycogen. Tamam. Do you guys remember the two linkages here?
Yes.
Yeah, okay. So the first enzyme that makes the alpha-1,4 is called glycogen synthase. Tamam. And the second one, which makes the carbon 1,6, is called branching enzyme. As it's here, you can see the enzyme glycogen synthase will make the linear form, and the branching enzyme will make the branches on the glycogen. Yes. So once we have the glycogen 6-phosphate, which is the first step in the glycogen synthesis, then we have to convert it to glucose 1-phosphate, and then we should give UTP to the glucose 1-phosphate in order to go to UDP glucose. You don't have to know this enzyme and these steps. Only know that glycogen synthase will make the linear form, which is alpha-1,4, and branching enzyme will make alpha-1,6. We're done with the glycogen synthesis. Let's go to breakdown. What do I mean by breakdown is we are now breaking down the linkages to give the body the glucose it needs. So if we broke down the alpha-1,4, we will get something called the glucose 1-phosphate, or when we break alpha-1,4 linkage, the primary thing or the product that we will get is glucose 1-phosphate, as we said here in this step. If we broke down the linear form, it will go back to glucose 1-phosphate. And if we need a free glucose, we should break the alpha-1,6 phosphatase, carbon 1,6. So we mentioned this before, phosphorylated glucose is trapped inside the cell. So if this is a liver and this is a glucose, the liver can dephosphorylate the glucose, and the glucose will be outside the liver and go to the blood. But if we have a muscle cell, the glucose is phosphorylated and it cannot go outside. So this is called trapped glucose. And this is really important. They can ask you about this in the exam. I remember 2 years ago, they asked us in the TBL about this trapping thing. In the liver, we have the glucose 6-phosphatase, which remove the phosphate out of the glucose. But in the muscle cell, we don't have it. This is why it's trapped. We have two new enzymes, which is the first one is debranching enzyme. It's self-explanatory, guys. What do you think it will do to the
It will break down the branch.
Yes, exactly. So debranching enzyme will break down this linkage, which is alpha-1,6. But we have another enzyme which is called the glycogen phosphorylase. Glycogen phosphorylase will break down the alpha-1,4, which is the linear form, and yes, it will give us the glucose 1-phosphate. In the liver, we have the glucose 6-phosphatase, which will make the glucose go to the blood, but in the muscles, we don't have it. So it will go from glucose 6-phosphate to glycolysis again. This is the enzyme that we mentioned. It will break alpha-1,4. It's called phosphorylase, and it removes the glucose molecules from the glycogen polymer. And the primary product is glucose 1-phosphate, as we mentioned before. These are the 4 enzymes that you have to know, and we took it before, but let's revise them because we will use them a lot in the next slide. For synthesis, we have glycogen synthase, which makes the linear form of the glycogen, and then we have the branching enzyme, which makes the branching glycogen. تمام. For degradation, we have the enzyme phosphorylase, which will break down the linear form, and for another enzyme, which is called debranching, it will break down the branching linkage and give us the free glucose. This is a checkpoint. Do you guys have any question? All clear?
Yes.
Okay, good. Okay, we have hormonal regulation, which regulate the glycogen metabolism. What do I mean by that is there is a hormones that makes the glycogen and facilitate the making of glycogen, and there is another enzymes that degrade the glycogen. We have 3 hormones that we have to know, which is first one is glucagon and epinephrine, and the third one is insulin. So what do you guys know about insulin, or what's the effect of insulin and glycogen metabolism?
I think insulin is secreted when the glucose in the blood is far beyond the threshold or the normal resting point.
Yes, exactly. So if we have a glucose in our blood and we have a lot of energy, do you think that insulin will enhance or will decrease the synthesis of glycogen? It will decrease the synthesis of glycogen because we already have a lot of energy. On the other hand, we have the glucagon and epinephrine. Glucagon will degrade the glycogen to glucose. When glycogen is secreted, when you are in a fasting state, when you are fasting at Ramadan or another day, and epinephrine. Why do you think, guys, epinephrine is will degrade the glycogen and will give us glucose? It will do that because it's secreted.
I think it has epinephrine is known as the flight or fight hormone, and when we're encountering such situation, we need energy. Therefore, excess glycogen is broken down to give us energy.
Yes, exactly. So when there's a lion is chasing you, you have to break down the glycogen by epinephrine, and it's secreted. So we will break down the glycogen by both glucagon and epinephrine. And on the other hand, insulin will decrease the degradation of glycogen. Let's start by glucagon and epinephrine. What they will do is they will decrease one enzyme and decrease the other enzyme. The first one, because they degrade the glycogen, they will increase the phosphorylase enzyme, which break down the alpha-1,4. Okay? They will not only increase the production of this enzyme, but will decrease the glycogen synthase, because glycogen synthase is the one that makes the alpha-1,4. So we don't need the body to make glycogen. We need to degrade glycogen in order to have those glucose monomers. And how they do this? By phosphorylating both of these enzymes. Phosphorylation does not mean we will increase the activity. Sometimes phosphorylation decreases the activity. So glucagon and epinephrine will phosphorylate both phosphorylase and glucagon synthase. But one, it will increase the activity of it, and the other one will decrease the activity. Is this part clear, guys? Or should I do it again?
I understood.
Okay, great. Glucagon and epinephrine, they don't directly phosphorylate both the glucagon phosphorylase and glycogen synthase. They have to go on a cascade steps. What do I mean by a cascade steps? Is it's a sequence of, sequence of events that phosphorylate the enzymes. First one is adenyl cyclase. Adenyl cyclase will give cyclic AMP, and from cyclic AMP, it will do the phosphokinase A, and phosphokinase A will phosphorylate everything. You don't have to remember this. Only know that epinephrine and glucagon will phosphorylate glycogen phosphorylase and glycogen synthase. And by phosphorylation, it will do the glycogen breakdown. For the muscle, and this is only in the muscle, it's not in the liver. The muscle contraction will use the calcium also to phosphorylate the glycogen phosphorylase, which will break down the glycogen. You don't have to know about the calmodulin. Only know that calcium will phosphorylate glycogen phosphorylase, and glycogen phosphorylase will break down alpha-1,4. Yes, and it will phosphorylate glycogen synthase, which will inhibit glycogen synthesis. Okay. The other hormone is insulin. We said that insulin is secreted when there is a lot of glucose in our blood. We don't need more glucose in the blood circulation. So insulin will decrease the glycogen degradation by 2 steps. First one, it will decrease the breakdown of alpha-1,4 by decreasing the phosphorylase, and it will increase the glycogen synthase activity, which will make the linkage between alpha-1,4. Is this clear? It's the opposite, but this time insulin will not phosphorylate any of these. And this is the picture to remember. Insulin does not need phosphorylation, so it will punch it down. This is the same thing. Insulin will go and bind, and then will remove the phosphorylation. So the glycogen synthase will start. You don't have to know this. And it will remove the phosphorylation as well from the glycogen phosphorylase. Okay. There's also something called allosteric regulation other than the hormonal regulation. Allosteric means we have molecules in our cells that will enhance the degradation or increase the making of glycogen. So if we have a lot of ATP and glucose, do you guys think we will make glycogen or we will degrade the glycogen? Which one? if we have a lot of ATP and glucose, this means no, we have a lot of energy. Will we make glycogen or degrade glycogen? Yeah, we will make glycogen because we already have a lot of energy. We don't need to degrade it. The other thing is AMP. What's AMP? It's ATP, but we took one of the phosphorylations. So it
We
becomes
took two of them out.
Yeah, two of them. So A and B means that we used a lot of ATP, we used a lot of energy. So now we have A and B, and when we used a lot of energy, we have now AMP, and AMP will start degrading glycogen and give us glucose, which is energy. And let's go back to the first part of the lecture. We will start synthesizing glycogen by the availability of glucose 6-phosphate. Is everything clear till now?
Yeah.
Okay, great. Now with the glycogen diseases, which is the last part of this lecture, we have something called, we have 6 types actually, from 1 to 6, they are numbered. We have something called McCurdley disease, which is caused by deficiency in myophosphorylase. What does myo mean? In medicine, myo mean muscles. And phosphorylase is the enzymes that break down the alpha-1,4 linkage. So if you have a deficiency in myophosphorylase, what do you guys think will happen in the muscle?
It will not, it will not take, no. The glucose will be given out, rather it won't be selfish anymore.
Yeah. We will not break down the glycogen in the muscle, so there will not be the anaerobic oxidation. Thus, we will not have enough ATP in the muscles. Tamam. If you are just walking, your muscles only need oxygen. But when you are when you are running for your life from that lion, the muscles need glycogen, and it will use the anaerobic oxidation. But if you don't have this enzyme, which is myophosphorylase, you cannot degrade the glycogen and you will not get glucose. Thus, the ATP will not be there. So the outcomes will be poor exercise tolerance and muscle cramps. You will be tired easily. And sugar before physical activity can do the work, which means it will enhance the outcomes, inshallah. The second one is type 2 disease, which is called bump disease. It's because there is a lysosomal enzyme deficiency, which is alpha-1,4 glucosidase. We did not mention all the enzymes in this lecture. We have another enzyme, which is alpha-1,4 glucosidase. This one also breaks down the alpha-1,4 other than the phosphorylase. So deficiency of this enzyme will cause accumulation in glycogen because we are not degrading any of them. So there will be accumulation in the organs like the heart. So the heart will be large, which is called cardiomegaly. And yes, it will cause, yes, never mind. And this is a summary of this lecture in one picture. And yes, do you guys have any question till now? Or should we do the quiz?
No question.
Okay, I will give you 2 minutes to read the question and type in the chat. C. Yes, correct. طيب, let's read the question. Summer is 13 years old. Who plays football? Football needs heavy exercise oxidation, which is it will your body have to break down glycogen and use the glucose because oxygen is not enough. So what you will have to have in your muscle is myophosphorylase enzyme. So what's this disease called? It's called McArdle disease. Okay, the second question. This should be two. B. Okay. Which of the following is correct regarding muscle glycogen? Glucose can be transported into the blood. This is X because the muscle don't have glucose 6-phosphatase enzyme. And C, muscle do not utilize glucose as a source of energy. This is wrong. It uses it when there is anaerobic oxidation or in intense exercises. So D as well is wrong. The answer is B. Glucose is trapped due to phosphorylation. Like, this is question 3. Sorry. Think again, Habibi. Which of the following is incorrect?
Yeah,
Which of
missed
the following
question.
false? Okay, so the answer is C. Insulin phosphorylate glycogen. No, wait a minute. Yes, insulin phosphorylate glycogen metabolism. It does not phosphorylate anything, guys. Remember this picture. Insulin will punch phosphor in the face because it does not have it. Only the glucagon and epinephrine will phosphorylate in order to activate and inactivate. Yes, and that's it. Do you guys have any questions before we end?
No. Thank you so much.
Thank you.
It was very well explained.
Thank you.
Have a nice day.
Habibi. Good luck in your exams, and if you need any help, just text me. خلاص?
Yes, thank you.
Thank you.